these molecules are going to act a lot like the rubber bands that I just showed you. So if you imagine then what cohesins look like, we need really to be able to entrap these two physical units of DNA. We need to be able to hold them together. We need some way that they're not going to come apart. And so if you, want to do that for some pieces of DNA, we're going to have some, some rings. Okay? And so what cohesin looks like as a complex is that there is going to be a long rubber band-like thing. This is going to be a region called a coiled coil, which is just a way to be able to spread things out and over long distances and maintain interactions. And so there's two proteins together that associate for that. And these are going to be referred to as SMC subunits. This is actually a protein that does also exist in bacteria, where it has slightly different structures and purposes. And then we're going to have another one of these same units come together here. Again, this sort of coil. These are going to associate with each other. And then we're going to need something to bridge this gap here. And we're going to have these things act to be able to be closed. And so we're going to refer to them again in an excellent salesmanship capacity as Cohesin subunits for close. Okay. And so there's a key protein that we'll come back to called SCC1 or RAD21, depending on the organism. And so this here is acting to be the closer. So you have the rubber bands itself, and you have a junction at the end, these Cohesins. And what these are going to do is actually just hold a piece of DNA in there. So here's one replicated chromosome, two strands of the DNA, and here it goes back behind that. And so it's coming out here. And similarly, now we're going to have the second replicated piece of DNA into that ring, back behind it, and over there. Okay. And so really, we've built a rubber band. We've really built a molecular rubber band. Here is the first chromosome. And here we have the second. Okay. This unit, these rubber bands, and I think currently the models for how we think about them, I think is pretty accurate in this way. These have to be placed on. And so these are loaded in S phase. So they're loaded during synthesis, really during the process of DNA replication. There are these rubber bands that also exist on chromosomes in other ways. So there's a version of that plays an important role in creating the organization that allows transcription during other phases of the cell cycle. We're really going to focus on the cohesin that is holding the two sister chromatids together, and that is loaded during this DNA replication process. And you imagine if we didn't load then, they're already separate. How do you get them back together? It would be a very, very hard thing to do. And so if we've lost that ability to hold them and lost it to load there, it would be problematic. One of the things you may have noticed, though, is that when you look at a picture of a mitotic chromosome, and even in those karyotypes, there was a pinching. And so often we'll draw a chromosome sort of like this, where a mitotic chromosome will look like that structure there. This is a mitotic chromosome. It's actually two replicated sister chromatids. So here's the first chromosome, and here's the second. But there's obviously this separation, and there's this sort of primary constriction here. This primary constriction is the centromere. This is the unit of DNA, the locus on the chromosome that is going to direct assembly of the machine called the kinetochore, which is going to hold on to the spindle microtubules and aid in their distribution. So the microtubules are going to come in there. That doesn't look at all like what you might expect from what I just told you. And so if we imagine, again, this chromosome, I've loaded the rings along the entire length. This does not look like what it is at all. And the reason for that is that a subset of these molecules, these cohesins down here on the arms, are actually going to be removed during early phases of mitosis. And so there's a machinery that includes a protein called WAPL that I'll write down in a second, which is going to leave the cohesin just there. And so we can actually build out a chromosome that looks much more like what you might expect from a chromosome spread, where it's held together with these cohesin molecules at the middle, but the edges and the periphery can separate from each other in different ways. So here we have our mitotic chromosome. So as much as we've built in the DNA, got to get a new piece of chalk now. We're going to remove arm cohesin. But we're going to protect it at the centromere. Okay. This is even better now. Now I think hopefully this again does look like a mitotic chromosome. But we're left with a problem, which is, again, let's pretend that I'm the spindle, my two elbows are the poles, my hands are microtubules. This black region here is the centromere. I can come in and attach that with microtubules. I can grab onto it. Now I'm holding them in opposite ways, and I'm going to pull on them, and we're going to come back to that later. And I can do this, but I'm not going anywhere because I have these ring-like molecules entrapping it. And so ultimately, I have to have a way to eliminate this, and eliminate this, I don't know, rapidly. I don't want them coming back, so I need it to be irreversible as well. And so one of the key themes that we talked about during the signal transduction lectures is that when we want to achieve a irreversible event, maybe even a rapid and irreversible event, we can do that by not just affecting the phosphorylation of these, maybe just not undoing them in the same way that WAPL molecule is, but let's come in with a protease. And so these scissors here are going to be our protease. We're going to hold on to these. And if I'm pulling in the opposite direction and I come in and I cut the rubber bands instead, now I can pull these apart. Okay? And so that's what the cell is going to do as well. And in fact, there's an excellent protease that's going to separate these two out. And again, we're excellent salesmen here. And so we're going to use a protease that's going to separate them. So it's going to be called separase. And separase is going to cleave the cohesin. It's going to really cut it. And in particular, it's going to cut the stuff on the chromosome arms, stuff at the centromeres that hasn't been removed yet. And it is going to specifically cut this RAD21 or SCC1 protein. And so separase is going to act on here. And it really is going to come in and it's going to break peptide bonds in these amino acids. It's going to break this RAD21 piece in half. When we do that, it's going to open this hole, and the rubber band can just fly off in the same way that I just described.